Friends, today we will learn about translation and regulation. This is the third video of translation and regulation. Let's know first of all proofreading. The fidelity of protein synthesis depends on the accuracy of the two mechanisms. The linking of each amino acid to its corresponding tRNA molecule and the base pairing of the codons in mRNA to the anticodons in tRNA. Two fundamentally different proofreading mechanisms are used in the cell. Both are active processing processes. One mechanism called chemical proofreading is used to improve the accuracy of amino acid attachment to tRNA. This is carried out by amino acid tRNA synthetase, which recognize an incorrect amino acid attached to its tRNA molecule and remove it by hydrolysis. A second mechanism called kinetic proofreading is used to improve the fidelity of codon-anticodon pairing. Once tRNA molecules have acquired an amino acid, they form a complex with an elongation into the growing polypeptide chain. The initial codon recognition, however, triggers the elongation factor to hydrolyze its bound GTP, whereupon the factor dissociates from the ribosome without its tRNA, allowing protein synthesis to proceed. The elongation factor thereby introduces a short delay between codon-anticodon base pairing and polypeptide chain elongation, which provides an opportunity for the bound tRNA molecule to exit from the ribosome. An incorrect tRNA molecule forms a smaller number of codon-anticodon hydrogen bonds than a correct one. It therefore binds more weakly to the ribosome and is more likely to dissociate during this period. Thus, the delay introduced by the elongation factor causes most incorrect bound tRNA molecule to leave the ribosome without being used for protein synthesis. Let's know about incorporation of Selenocysteine resembles cysteine but contains selenium instead of sulfur. Selenocysteine is encoded by UGA. However, UGA is one of the stop codons. Apparently, UGA is normally read as stop but is occasionally translated to give selenocysteine. The choice between stop and selenocysteine depends on a special recognition sequence called selenocysteine insertion sequence. Selenocysteine has its own tRNA and special protein factor to escort charged tRNA seq to the ribosome. In fact, selenocysteine tRNA is initially charged with serine. Then the attached serine is enzymatically modified to form selenocysteine. When bacteria use selenocysteine, the selenocysteine insertion sequence forms a stem and loop structure in the mRNA molecule just after the UGA. Cell B protein recognizes both charged tRNA seq and the stem and loop. Thus, selenocysteine bound to tRNA is delivered to the right place. Sex-specific tRNA in presence of serine and serine tRNA ligase gives rise to tRNA cell gives rise to seq synthetase in presence of seq synthetase gives rise to tRNA seq. Now come to cap snatching. Some viruses such as influenza virus perform a unique cap snatching process. Cap snatching is a transcription initiation process during which a nucleotide sequence between 10 to 13 is in size is cleaved from the 5' end of the host mRNA by an endonuclease activity present in the viral RNA dependent RNA polymerase. The capped nucleotide sequence removed from host mRNA's 5 is subsequently used as a primer for transcription of the viral genome, which ultimately leads to the synthesis of capped viral mRNAs. Now let's know about translational frame shifting. Translational frame shifting is a mechanism by which the translational machinery shifts the frame in which it decodes the mRNA. The result is that the mRNA does not encode the protein by a continuous run of three nucleotide codons known as an open reading frame, that is ORF. Rather, the information encoding the protein comes from two distinct ORF5. Frame shifting is a sto- stochastic process, meaning that each translating ribosome has a certain probability of undergoing the shift, but that only a fraction of the ribosome does so. Classes of signals have been identified that direct a fraction of elongating ribosomes to shift reading frame by one base in the 5' or 3' direction. In general, it is believed that the occurrence and frequency of translational frame shifting are determined predominantly by two elements of mRNA, a slippery sequence and a downstream RNA structure. Various slippery sequences have been identified from different retroviruses and from other viruses. A typical slippery sequence is a heptanucleus nucleotide X X X Y Y Y Z, where C X equals to B, X equals to C, B equals to A, or U and Z single bond G. There are two kinds of downstream RNA structures associated with frame shifting: stem loop and pseudo knot. However, not all stream loop or pseudo knot structures can induce ribosomal frame shifting. Thus, the definitive characterization of specific RNA structures and how these RNA structures mediate ribosomal frame shifting remain to be defined. Ribosomes that frame shift produce a translational fusion of the two overlapping ORFs, whereas those that do not frame shift continue normal in frame decoding and terminate at the end of the first ORF. Each of these products thus shares a common N-terminal region. Many viruses use programmed translational frame shifting to ensure synthesis of the correct ratios of virus encoded proteins required for proper viral particle assembly and maturation. The phenomenon was first described in year 1985 as the way in which gag fall polyprotein of the retrovirus Ross sarcoma virus RSV is expressed from the overlapping GAG and POL ORFS. It has been demonstrated that when ribosomes translate the unspliced genomic RNA of retroviruses, 95% of translation yields GAG proteins, while only about 5% of translation produces GAG pol proteins through -1 ribosomal frame shifting. Now come to antibiotics and toxins. Protein synthesis is a target of a wide variety of naturally occurring antibiotics and toxins. Mechanism of action of some common antibiotics and toxins which inhibit protein synthesis in prokaryotes and eukaryotes are. Let's know about some antibiotics and toxins. Streptomycin. Streptomycin is a basic trisaccharide binds with the 30S subunit of the bacterial ribosome and causes misleading of mRNA at relatively low concentrations. Now come to chloramphenicol. Chloramphenicol binds to the 50S ribosomal subunit and blocks peptide bond formation through inhibition of peptidyl transferase but does not affect the cytosolic protein synthesis in eukaryotes. Tetracyclines binds to the 30S ribosomal subunit and interferes with amino acid tRNA binding. Erythromycin. Let's know about erythromycin. Erythromycin binds to the 50S ribosomal subunit and inhibits peptide chain elongation. Now come to fusidic acid. Fusidic acid binds to EFG and blocks translocation. Cycloheximide. Cycloheximide blocks the peptidyl transferase of FTS ribosome, but not that of 70S bacterial ribosomes and mitochondrial and chloroplast. Puromycin. Puromycin is a secondary metabolite of streptomyces alboniger that blocks protein synthesis. Puromycin is a structural analog of the 3' end of amino acid transfer RNA but differs from tRNA insofar as the amino acid residue is linked to the ribose via an amide bond rather than an ester bond. Puromycin, like amino acid tRNA, binds to the A site of the ribosome peptidyl transferase center. When the when the A site is occupied by puromycin, peptidyl transferase links the peptide residue of the peptidyl tRNA in the ribosomal P site covalently to puromycin. Since the amide bond cannot be cleaved by the ribosome, no further peptidyl transfer takes place and the peptidyl puromycin complex falls off the ribosome. Now come to diphtheria toxin. Diphtheria toxin, an exotoxin of Corynebacterium diphtheria infected with a specific lysogenic phase, stops the protein synthesis in eukaryotes by inactivating the elongation factor E small e capital F 2. Elongation of factor E capital F2 occurs due to ADP ribosylation, which is catalyzed by a fragment of toxin. Now come to ricin. Ricin, a toxic protein of castor bean, Ricin cuminis, that inactivates the 60S subunit of eukaryotic ribosomes by depurinating a specific adenosine in 28S RNA. Now come to post-translational modification of peptides. Chemical modification. Primary translation products often undergo a variety of modification reactions involving the addition of chemical groups which are attached covalently to the polypeptide. This can involve simple chemical modification like hydroxylation and phosphorylation of the side chains of single amino acids or the addition of different types of carbohydrate or lipid group. Example of Let's know about some examples of post-translational chemical modifications. Amino acids that are modified. Let's know first is modification, next is amino acids that are modified. First is acetylation, LYS, methylation, LYS, phosphorylation, SIR, SER, 309, THR, tyrosine, TRY, aspirin, ASP, histine protein, HIS, lysine, LYS. Now come to thank you. Let's know about proteolytic cleavage. Proteolytic cleavage of peptide chains after synthesis is a common occurrence with certain classes of proteins. For example, proteolytic enzymes in the digestive tract are produced in inactive forms that are generally termed as zymogen. After selective proteolysis, these enzymes are converted into active forms. One best known example of proteolytic cleavage is the conversion of the pre-pro-insulin into insulin. First, removal of the signal peptide from the newly synthesized peptide pre pre-pro-insulin generates the pro-insulin precursor molecule. Finally, the removal of C peptide moiety of proinsulin gives insulin. Now come to protein splicing. Occasionally, the primary translation product of a gene contains one or more short amino acid sequences called intents that excise themselves from the nascent polypeptide. The sequences that are presented in mature polypeptide are termed extins. Intins occur in both eukaryotic and prokaryotic polypeptides. An intin has the ability to catalyze its own removal from primary translation products. The autocatalytic process of protein splicing involves bond transfer reactions and no input of energy is required. According to the most accepted theory, the standard protein splicing mechanism consists of four steps. Let's know about these steps. First step is NO shift. A typical intin begins with SIR or CYS and ends in ASN. The first residue in the C16 is SIR, THR or CYS. These residues often function as nucleophiles. NO shift. NO shift is the transition of the peptide bond between the amino end of the intent and N extend into an ester or thioester bond. This transition depends on a nucleophilic attack of the bond by the side chain of the SIR or cis residues at the amino terminal end of the intine. This reaction is termed NO when the attacking atom is an oxygen and NS when this atom is sulfur. Second step. Let's know about the second step. The second step is transesterification. In this step, the side chain of the first residue of the C extend attacks the ester bond at the amino acid end of the intend. Here, too, the attack is by a polar side chain of a SIR, THR, both OH or cis SH. This leads this leads to the esterification, transesterification and formation of thioester or ester bond between N-histine and C-histine. Now let's know about third step. Third step is ASN cyclization. Cyclization of the ASN side chain leads to cleavage of the peptide bond between the intine and the C-histine, that is C-terminus splice junction. This reaction removes intine from the ligated extins which are linked together via the ester bond. Now come to fourth step. Fourth step is ON shift. This step of protein splicing is spontaneous. The reverse NC or NS shift takes place and peptide bond formation occurs between N and C extins. Some intins show sequence-specific endonuclease activity also. Such intins cut DNA in the intin minus gene at the specific point and allow a copy of a DNA sequence coding intin to integrate. This event is similar to intron homing and termed as intin homing. To ubiquitin mediated protein degradation. The ability to degrade proteins is an essential function of all eukaryotic cells. The two main proteolytic systems within eukaryotic cells are lysosomes and proteasomes. Lysosomes are membrane-bound organelles that contain an assortment of acid hydrolyses, including many proteolytic enzymes. Proteosomes are cylindrical, multi-subunit proteases found in both prokaryotes and eukaryotes. In eukaryotes, they are present in both cystole and the nucleus. Eukaryotic proteosomes are of two types, 20S proteasome and 26 proteasome. The 205 proteasome is a cylindrical, barrel-shaped structure consisting 0, 1 to 8 subunits arranged as four rings of heptamers stacked upon one another. All subunits are the members of the same superfamily of proteins which group into two families designated alpha and beta. In eukaryotic cells, the 20S proteasome assembles with two 19S regulatory complexes RC in an ATP dependent manner to form the 265 proteasome. Now come to ubiquitination or ubiquitinal of substrates. The most well established means of targeting proteins to proteosomes is by their modification with chains of ubiquitin. Ubiquitin, a highly conserved eukaryotic protein of 76 amino acid residues, is usually attached to substrates by an isopeptide bond between a substrate lysine residue and a C-terminal glycine of ubiquitin. The process of ubiquitinylation occurs in various steps. Let's know about the steps. First is activation of ubiquitin. Ubiquitin is activated by an E1 ubiquitin activated enzyme. E1 becomes covalently linked to the free ubiquitin through the free C-terminal residue of ubiquitin in an energy dependent manner. Now, second is transfer of ubiquitin from E1 to E2. The activated ubiquitin is subsequently transferred to a cysteine residue present on an E2 ubiquitin conjugating enzyme. Ligation, third point is ligation, third step is ligation of ubiquitin to target protein. Finally, E3 ubiquitin ligases transfer the activated ubiquitin from E2 to a lyse amino acid residue of its target protein, forming an isopeptide bond. A ubiquitinated protein is proteolytically degraded to short peptide in an ATP-dependent process mediated by proteasome. Thank you.